Welcome to this explainer. Today, we're taking a deeply complex immunological breakthrough and turning it into, honestly, a pretty captivating story. We're looking at a future where doctors might actually stop organ rejection entirely, not by nuking the immune system, but by simply disarming our own immune cells. It's a fascinating journey into the microscopic world inside us. Let's get right into it.
Okay, here's our roadmap for today. We'll be hitting the transplant rejection problem, calcium as the immune trigger, testing in the lab, testing in living mice, and finally, a future for transplants.
Starting off with part 1, the transplant rejection problem. To really grasp the stakes here, let's quickly get our terms straight. When a patient receives an allograft, a tissue or organ from a donor with different genetics, the body's lymphocytes, its immune cells, instantly spot it as an invader and launch a deadly attack. But there's a flip side, graft-versus-host disease, or GVHD. This is a severe, sometimes fatal scenario where the immune cells hidden inside the new organ wake up and actually attack the recipient's body. Both of these nightmare scenarios boil down to one basic issue: mismatched immunity. Now look at the stark contrast between how we handle this right now versus the dream scenario. Our current method is basically a sledgehammer. We use harsh, whole-body immunosuppression. We shut down the entire immune system just to protect the organ. But as you can imagine, that leaves the patient totally defenseless against everyday bugs. The ideal solution? Specific tolerance. Researchers are chasing a precise, targeted ceasefire where only the specific lymphocytes reacting to the new organ get neutralized. The rest of the immune system? Perfectly intact and ready to fight off real threats.
Moving on to part 2, calcium as the immune trigger. When you hear calcium, you probably think of a glass of milk and strong bones, right? Well, in a landmark 2005 study published in Immunology Letters, researchers Mohan Saxena and Madhu Dixit went a totally different route. They looked at a bold new hypothesis. They wanted to tackle graft rejection not by looking at the whole organ, but by zooming way in on what's happening inside the individual immune cell during an attack.
They zeroed in on a crucial mechanism. Think of lymphocytes as the body's internal army. Well, intracellular calcium, that is their green light to attack. When these cells detect foreign tissue, calcium levels inside the cell absolutely spike, signaling the cell to multiply and go on the offensive. So the aha moment here is that by blocking this intracellular calcium, doctors wouldn't have to kill off the immune army. They could literally just take away their weapons, stopping the attack signal before it's ever even sent.
To put this to the test, the researchers brought in 2 highly specific chemical heroes designed to block calcium but in completely different environments. First up is BAPTA-AM, an intracellular calcium chelator. This binds to calcium and was used to treat donor cells in vitro, meaning in a lab dish. Then we have KMB8, a calcium antagonist. This one was used for treating the host in vivo, meaning inside a complex living body.
All right, part 3, testing in the lab. Check out this stark difference from their first trial involving mouse lymphocytes. The untreated control group had a massive 90.05% of their lymphocytes activated, totally ready to wage war. But when they hit them with the calcium blocker BAPTA-AM, that activation plunged down to 58.81%. That is a highly significant, verifiable drop in cellular aggression just by tying up the calcium inside the cells. And what this tells us is incredibly promising. These lab results proved that by basically starving the cells of calcium, you can severely cripple the aggressive immune response against foreign tissues. This means in vitro treatment successfully disarms the donor's immune cells before the organ ever even touches the new host. They're potentially neutralizing the threat of graft-versus-host disease right there in the petri dish.
Which brings us to part 4, testing in living mice.
As we all know, petri dishes are not people. The next critical step was seeing how this chemical intervention plays out in an actual, living biological system. So the researchers measured something called delayed type hypersensitivity, or DTH. They did this by injecting a stimulant into the foot pads of sensitized mice. In a normal immune response, lymphocytes rush to the site to attack, causing the foot pad to swell up. By measuring that swelling, the researchers could literally see exactly how aggressive the immune system was being. So they treated these mice with that living body calcium blocker, TMB8. A low initial dose had a pretty mild effect, but just look at what happened when they upped it to a 0.1 milligram dose. The swelling was exactly 0.000 millimeters. The physical immune response was completely abrogated. I mean, this is a stunning result. TMB8 entirely neutralized the swelling. The immune army had the foreign stimulant sitting right in front of them, but because their calcium channels were blocked, they simply stood down.
But hey, biology is rarely that simple, right? The researchers also measured the internal mixed lymphocyte reaction, or MLR. The untreated naive mice never exposed to the foreign tissue, sat at a baseline of about 59.6% activation. Mice transfused with mismatched cells spiked to 72.8%. But here's the surprise. The TMBA-treated transfused mice actually sat at 76%. So while TMBA stopped the physical swelling dead in its tracks, the internal activation magnitude in treated mice stayed comparable to the untreated ones. It really highlights the sheer complexity of whole-body immunity. It tells us that even though the physical attack is halted, the cells are still dynamically interacting with the antagonist behind the scenes.
Finally, Part 5, A Future for Transplants. Based on all this data, the researchers proposed a powerful two-pronged strategy for future organ transplants. Step 1. Disarm the organ in a dish. You treat the donor lymphocytes in vitro with a chelator like BAPT-AM so the organ doesn't attack the patient. Step 2. Gently calm the patient's immune system. You treat the recipient in vivo with an antagonist like TMB8 so the patient's body doesn't reject the organ. This combined strategy, pioneered by brilliant researchers like Dr. Saxena, is an absolute paradigm shift. The goal is to protect the new organ from the body, and the body from the new organ. And the best part? It aims to do all of this without the sledgehammer effect of destroying the patient's overall health and leaving them wide open to other diseases. The authors boldly state this in their conclusion, and honestly, this quote is the heart of the whole thing. They say this specific targeting of calcium inhibitors could possibly lead to acceptance of mismatched allografts by an immunocompetent recipient. Think about that for a second. An immunocompetent recipient. That means a patient with a fully healthy, completely functioning immune system who calmly and peacefully accepts a mismatched, life-saving organ. That's the holy grail. Thank you so much for exploring this fascinating study with me today in this explainer. It's just incredible how looking at something as microscopic as calcium inside a single cell can completely upend the macro world of medicine. And it leaves us with an incredibly provocative question. Are we truly on the verge of a world where waiting years for a perfect match organ is a thing of the past? Because if calcium inhibitors can bridge the gap of mismatched tissues, the future of transplant medicine might be a whole lot closer than we think.